In the mid-20th century, the world was rebuilding from war. But in the quiet corridors of science, another race was on. Researchers across the globe were sprinting towards a monumental prize, unlocking the structure of DNA, the very molecule that holds the blueprint of life. The answer, they knew, would change biology forever. Amid this scientific frenzy, one brilliant mind at King's College London was closing in on the truth. Her name was Rosalind Franklin, and she was a master of one of the most challenging techniques in science, X-ray. This method wasn't like a normal microscope. Instead, it involved firing a beam of X-rays at a crystallized molecule. As the rays scattered, they created a pattern, a shadow of the molecule's inner architecture, revealing its secrets in a way no one had ever seen before. Franklin's work was a testament to her precision and relentless dedication. For months on end, she labored in her lab, painstakingly preparing delicate fibers of DNA and refining her equipment to achieve unprecedented clarity. This wasn't a quick process. Each exposure required immense patience, with some experiments running for over 100 hours just to capture a single image. But her persistence paid off. One day, after developing a plate, an image emerged that was clearer and more revealing than any before it. Labeled simply Photograph 51, it was a masterpiece of experimental science. The image showed a distinct, cross-shaped pattern, an unmistakable X at its center. To a trained eye, this was the smoking gun, the signature of a helix. It was the most crucial piece of physical evidence yet uncovered in the quest to understand life itself. This is where the story takes a dramatic and controversial turn. Unbeknownst to Franklin, a colleague, Maurice Wilkins, showed Photograph 51 to two competing scientists from Cambridge University, James Watson and Francis Crick. For Watson, seeing the image was a revelation, a sudden and brilliant flash of insight. The distinct X pattern was the missing piece of their theoretical puzzle. It immediately confirmed their suspicion that DNA was a helix, and more importantly, gave them the precise measurements they needed to figure out how the whole structure fit together. Armed with Franklin's data, they were able to rapidly assemble their now iconic model of the double helix, correctly deducing the final, elegant structure of the molecule of life. In April 1953, Watson and Crick published their groundbreaking paper in the journal Nature, announcing their double helix model to the world. It was a landmark achievement that would reshape science. In the very same issue, Rosalind Franklin's own paper was published, complete with the critical data from Photograph 51. However, it was positioned as mere supporting evidence for Watson and Crick's theoretical triumph, rather than the foundational discovery it truly was. Tragically, Franklin's story was cut short when she died of ovarian cancer in 1958 at the age of just 37. 4 years later, the Nobel Prize for the discovery was awarded to Watson, Crick, and Wilkins, with her name and pivotal role largely relegated to a footnote. Today, we remember her not as a supporting character, but as the unsung heroine of this story. Rosalind Franklin's experimental genius provided the key that unlocked the secret of life, securing her rightful place as a pioneer of modern biology.